Hi, everyone. We're back again, and this section of the lecture is on a different type of a different regulatory system in bacteria. Um, it uses a fairly standard activator with with an effector molecule, but the interesting thing about it is that the effector molecule is actually produced by the bacteria themselves. And it is allowed to be released from the cells, and it's used essentially as a density sensor. So the cells, the bacterial culture can keep track of its population. So this system is referred to as quorum sensing. Now, those of you who were in Bio 1C in the last year or two have already learned about this. And those of you who weren't are just going to review it again. If you have learned about it, then this will be just a refresher to you. But I want you to think about this now from the perspective of the sort of regulatory strategy. So let's see, where to start. So I said it's referred to as quorum sensing. It's used to monitor population, and it has to do with a release of a factor called the autoinducer. So let's go on to the next slide here. All right, so this was discovered in bacteria that live in a symbiotic relationship with fish and squids. And the amazing things about these bacteria is that they produce light. They are bioluminescent. The bacteria is called Vibrio fischeri, or there are other species as well, Vibrio harveyi. These are from the same genus of bacteria as Vibrio cholerae, the bacteria that causes the disease cholera. But these bacteria live in harmony or in symbiosis with their host, providing a service to the host, in return receiving food from the host. All right, so the system is called the Lux system. Lux for sort of shorthand for luciferase, which is the enzyme that produces the light in these bacteria. All right, so the bacteria can live in the open ocean, or they can live in symbiosis with the squid, but they only produce luciferase and light when they are living in the symbiotic relationship at high density. So the question is, how do the bacteria know when to produce the light? Before we answer that, we might ask what the squid actually use the light for. And I should say this is just a theory. And as relates to the squid, and this is a simulation on the left here, that the squid produce light in the light organ so that a predator looking at them from the bottom on a moonlit night, near the surface, would not be able to see them as well. I've always found this to be somewhat of a limited, or I'm skeptical that this is a true use. Nevertheless, the symbiosis evolved, and so what can we say? So the bacteria live in an organ within the mantle of the squid called the light organ. And while they are there, they receive food from their host. They proliferate to high densities, and once they reach higher density is when they turn on light production. Okay, so how do they know they have reached high density? The signaling molecule that the bacteria produce is of a chemical family you see down here. These are called N-acyl homoserine lactones. Right, I'm not going to bother going through the structure, and the structure is variable depending on the species that it's coming from, because as we'll talk about in a little while, this regulatory mechanism has been adapted to a lot of uses, not just for controlling luminescence in symbiotic marine bacteria. Okay, so the autoinducer is sensed as a signal to stimulate luminescence, and it does this by binding to a transcriptional activator called LuxR. The protein or the enzyme LuxI is actually what catalyzes the final step in the synthesis of the acyl homoserine lactone. And the idea is that at low cell density, the bacteria are producing this AHL, but it doesn't build up to high enough levels to turn on light emission. I'll show you that next. All right, so this is the sort of the details of the operon and the regulatory strategy. So we have the Lux operon here, and then we have the regulator here. This is similar to how the LAC operon is organized, where the genes that are regulated are closely adjacent to the gene for the actual regulator, in this case, the transcriptional activator, okay? So Lux I is here. This catalyzes the final step in AHL synthesis. And then in the rest of the Lux operon, we have genes that are involved in making the substrate that luciferase sort of burns through, in a sense, and then Lux A and Lux B, which are the subunits of the luciferase enzyme itself, okay? So this promoter here has an operator associated with it called the Lux box, and the RNA polymerase will bind to the promoter and express it at low levels. This is kind of like the LAC operon promoter in that it's a fairly weak promoter by itself, and this LuxR regulator is going to activate it to turn on the expression. Okay, so the substrate for, or the effector molecule for LuxR is this AHL. Now, when this operon is expressed at low levels without the participation of LuxR, it makes this AHL. You can think of this as sort of the constitutive level of AHL. But the thing about AHL is that it's fairly nonpolar, and it can diffuse across the membrane. If I could go back here, I don't seem to be doing a very good job of going back. Let me stop this for just a second and sort of regain control of my PowerPoint here. All right, I'm going to share the screen with you again. Sorry about that. There we go. Go back here is what I wanted to. This is a fairly nonpolar molecule. So the point is that it can diffuse across this cell membrane fairly readily. Okay, so essentially AHL is produced inside the cell. It's equilibrating across this membrane. Okay, so when there's very little of it out here, essentially it's leaking out of the cell. So there's very low concentration in here. Okay, now, at high concentrations, when there's a lot of these bacteria around, all of which are producing, their constitutive level of AHL, and these are in the light organ of the squid, I should point out. The AHL is slow to diffuse out of the light organ because there's a lot of tissue for it to go through. So as these bacteria build up here, concentration of AHL builds up as well, okay? And so as we said, it's equilibrating across the membrane, which means that the concentration of AHL inside the cell is increasing as well, which means that it binds to LuxR, which means that LuxR then binds to its operator and activates the RNA polymerase to express this gene, okay, or this operon. And so that makes more luciferase, it makes more of the substrate for luciferase, and it makes more AHL. This is sort of a classic positive feedback loop, okay? So as it gets turned on, it makes more of the signal to turn it on at even higher level, etc. Right? So rather than just having a linear increase in the light production, you get a dramatic sort of a threshold at which this positive feedback loop kicks in, and you get a sudden dramatic increase in light production. So that happens between like 10 to the 8th and 10 to the 9th CFU per mil of the vibrios inside the light organ. So when they start to build up, all of a sudden it turns on because of this positive feedback loop. All right. So hopefully you get the idea then of how this works. Now, there's been a lot of research done in the last decade or two since this was discovered as to the relationship between the squid and the bacteria and how the squid sort of nurtures the bacteria. The other thing you may remember, and I'm going to put a link to a nice video on quorum sensing that you may remember from Bio 1C, is that the squid actually expels most of the bacteria from the light organ during the daytime when they're not needed. And then the remaining bacteria don't build back up to high levels until basically the nighttime again when they turn on light production. So the squid is not bothering to keep these bacteria at very high concentrations all the time. It's essentially booting them out, letting them grow back up again, using them during the night, booting them out again the next day, etc. Okay, so when the bacteria get booted out of the light organ, now they're out there in the great big ocean. The concentration of the bacteria has dropped dramatically, and so the AHL concentration drops dramatically, and they turn this back off when they're in the free living state. So it's only on when they're in that symbiotic relationship. All right. So what's been learned in the years since this quorum sensing was discovered in the symbiotic light producing bacteria is that this system is used for actually a lot of different things in different bacteria. Mostly in gram-negative bacteria, it's used to control things like biofilm formation. When the bacteria start growing densely on a surface, they sense that there are lots of other cells around and they adjust their behavior, turn on the extracellular polysaccharide formation, etc. It's used in sensing when they're in the host in a pathogenic condition by Vibrio cholera, a close relative of these symbiotic Vibrios, etc. It's used to control conjugation because you don't want to, need to be making conjugal pili and stuff when there's no other bacteria to conjugate with, right? So quorum sensing is turned into a really interesting field of study because it's used for a lot of different things. Turns out that gram-positive bacteria use quorum sensing, but they don't use the same mechanism. They usually secrete peptides that they then sense on the surface of the cells as well. So it's a way of sensing population, is just not mechanistically the same as what we were just talking about. Okay, so super interesting. All right, I'm gonna stop this video now. Stop sharing this, and I'm going to stop, and then there will be another video wrapping up this section.